Hello, everybody. In this new unit, we are going to take our deepest dive into molecular biology and look deep inside of the cell to see what molecules are doing to carry out life processes. We're going to focus on cellular respiration and photosynthesis, two of the most fundamental metabolic processes. But in order to understand them, we first have to understand enzymes. So let's get started. Enzymes are proteins. And once you hear that they're proteins, then you know that the only way they can be produced is through transcription and translation. So there are genes for these enzymes. They are proteins that facilitate the chemical reactions in the cell and other parts of the body. So why is this necessary? Well, it's necessary because at the ambient conditions within the cell, there is normally not enough energy available for the reactions to happen at a significant rate. So the enzymes then are able to speed up the rate of the reaction, which allows cells to maintain themselves at the temperature that we find on Earth. The enzyme decreases the activation energy required to get the chemical reaction to occur, and I will explain more about that in detail later. Once the reaction occurs, the enzyme releases the product that it has made, and it is then able to facilitate another reaction. This ability to speed up a reaction while not being changed by the reaction makes enzymes a catalyst. As a protein, the enzyme has a three-dimensional shape that is necessary for it to function. We've talked about this all along. When we talked about transcription and translation, we talked about enzymes that needed to be transported to the Golgi apparatus, where they are further processed and folded into proper shape. And now we're going to see one of the reasons why. The exact shape of the enzyme is known as its conformation, and it is key to the enzyme's ability to catalyze a reaction. The substrate, also known as the reactants, attaches to a specific location on the enzyme called the active site, and I'm going to show you that in the next slide. Here we see the enzyme Sucrase, which is going to take the molecule sucrose, which is a disaccharide, meaning it's composed of two separate sugars. Sucrose, you'll remember, is table sugar, and break it down into its two component sugars, glucose and fructose. Let's see how that works. The sucrose binds to the active site, and then using water, the enzyme breaks the sucrose into two separate sugars, fructose and glucose. They are released, and the enzyme is able to work again. It's important that you realize that enzymes do not make a reaction occur that would not occur on its own. They just make it happen faster. Enzymes don't do magic. They do reactions that would occur naturally, but at a much slower rate. It's also important to know that enzymes are highly specific. In general, they will catalyze a single type of chemical reaction for one or two, and maybe rarely three substrates. And we refer to this as enzyme specificity. Enzymes are highly specific in the substrates they can act upon. Enzymes are really fast, often on the order of millions of reactions a second. And the reactions are usually reversible, meaning the enzyme can catalyze the reaction in either direction. But it usually only does so in one direction because the products are being used up by another enzyme, and so they continue in the direction that they were going rather than reversing. There are about 2,700 types of enzymes in the human body, which means about 2,700 genes for enzymes. We can think of them as carrying out four types of services in the cells. The first is they build structures such as cytoskeleton, external skeleton such as bone in mammals, and exoskeleton in arthropods, which are things like lobsters, crabs, and insects. They also can be molecular motors, which we saw earlier. They can also be digestive enzymes, and they break down food particles into simpler molecules, and they can do that outside the cell as they do in our digestive tract, or inside the cell as they do in lysosomes. And finally, they can carry out what I refer to as basic metabolism, which are the chemical processes necessary to keep the cell alive. Things like cellular respiration, photosynthesis. In a sense, all enzymes carry out metabolism because the building of structures, molecular motors, digestive enzymes are all necessary for keeping a cell alive. But I still think this is a useful way of thinking about different types of functions that enzymes have. An enzyme's activity can be affected by general environmental factors such as temperature and pH, and molecules that specifically influence the enzyme, both positively and negatively. And we will discuss these later on to show how cells control the rate of enzymatic reactions. For now, let's focus on temperature and pH. These two variables may change the shape, the conformation of an enzyme. And if they change it to a form in which the enzyme can't act, we call it denaturation. So cooked egg whites are proteins that have been denatured. Ceviche, which is fish that has been cooked in lemon juice, the very high acidity of the lemon juice denatures the proteins and makes them more easily digestible, are examples of denaturing by temperature and pH respectively. Because the shape of the enzyme, specifically the shape of the active site, is so important to an enzyme's ability to function, anything that can change its shape will reduce or eliminate enzyme function. pH, which is the concentration of the H+ ion, and temperature affect the shape of the protein. And as a result, each enzyme has an optimal temperature in which it can function, and each enzyme has an optimal pH in which it can function. Here are two enzymes, one that is found in a human, and you can see its optimum is in the high 30s, which happens to be the average body temperature of a human. And here is an enzyme from an archaean, which lives in a hot vent in the ocean, and you can see that the temperature optimum for its enzymes is way up near 80 degrees C, very hot. At 80 degrees C, our enzymes would cease to function. And what this means is, as you get away from these temperatures, the ability of the enzyme to function, the rate of the reaction, and that's how quickly product is formed, decreases as you go away from the optimum. Here is a similar graph for two digestive enzymes. One is called pepsin, and it is found in the stomach where the pH is low, and not surprisingly, its optimum pH for digesting protein is just around 2. And trypsin, which is an intestinal enzyme that digests protein, has an optimum about 8 because the pH of the intestine is raised to about pH 8. And so you can see these two enzymes have evolved to have different pH optima. Now, I want you to think about the rate of chemical reactions for a second. Here is an old book. It can be read hundreds of years after it was made, and yet it can be destroyed in an instant if it is lit on fire. However, this book has deteriorated slowly. So how can we account for these aspects of paper, that it can deteriorate slowly or it can deteriorate rapidly with fire? The first thing to realize is that both the slow destruction with age and its destruction by combustion are the same chemical process in which oxygen is reacting with the paper and deteriorating it, changing its chemical structure. To understand what's going on here, we have to think a little bit about chemical reactions. So chemical reactions consist of the breaking and reforming of bonds between atoms and molecules. For example, this diagram of the equation of photosynthesis shows that the molecules water and carbon dioxide are broken and rearranged into one molecule of glucose and 6 molecules of oxygen. That's a chemical reaction. The same number of hydrogens, carbons, and oxygens are on both sides and are accounted for both before and afterwards, but the molecular structure of the products and the reactants has been changed. That is a chemical reaction. All chemical reactions require an input of energy to cause the reaction to begin. In other words, in order to activate the reaction, we have to put in energy, and so we call this an activation energy. And here is a picture of this idea. Here we can see that the reactants, which start with a certain amount of energy, require an input of energy and then will form the products, which have a lower energy than the reactants. Similarly, some reactants require a large amount of energy and create products that have a high amount of energy in them. So some products are less energetic, and so the reaction gives off heat here, and sometimes the opposite is true, where the reaction absorbs heat. The activation energy, that initial energy needed to start a chemical reaction, can therefore be viewed as the thing that prevents chemical reactions from occurring at low temperatures. In other words, it provides stability for chemical compounds. Why doesn't the paper in this book spontaneously catch on fire? Because it takes some activation energy to get it to react with oxygen and break down. That's why the paper doesn't burn spontaneously. But if you use a match, that match gives it sufficient energy, and then the book will burn in my hand and will continue to do so because the match provides the initial energy, and then the energy released by the burning of the paper continues to provide enough energy to keep that chemical reaction going. You might be wondering, how do enzymes lower the activation energy barrier? What they don't do is they don't affect the change in energy between the reactants and products. Instead, they hasten the reactions. And this answers the question, why does the book deteriorate over time? The answer is that the chemical reaction of the paper with the oxygen still occurs, but it occurs very, very slowly. In other words, in the absence of the match or the activation energy, the book does a slow burn over time, especially if it gets you angry. How do enzymes lower the activation energy? We still want to know that. And they do it by several things. They do it by orienting the substrates correctly. In other words, if there's two substrates, they hold them in just the right way, so they increase the likelihood that they'll react. They strain the bonds in the substrates physically, they twist them. They can provide a favorable microenvironment, such as a high or a low pH. And they can actually bond to the substrate briefly and change the reactivity of the electrons in the molecules. And so they make it more likely that the chemical reaction will occur. And here's a picture of that. Here we see that without an enzyme, the activation energy is given by this arrow here, but with an enzyme, the activation energy is lowered. Notice that the energy contained in the reactants and the energy contained in the products is not affected by the enzyme. That's it for today. What I hope you understand today is that enzymes are proteins and therefore coded in the genes, that they act as catalysts, a substance that increases the rate of a chemical reaction without itself being affected by the chemical reaction, and that enzymes are important for metabolism, the chemical reactions that allow a cell to remain alive, that there are external factors that affect the ability of an enzyme to carry out its reaction. The two most important are temperature and pH. And that the way enzymes work is by lowering the amount of energy required to get a chemical reaction to occur that will occur naturally, but at a slower rate. So until next time, go outside or pick up a biochemistry book, take a look around or read it, ask biological questions and bring them back to class, and I'll see you soon.